Learning outcomes. After studying this module, you shall be able to know some important instruments and technique used in forensic science. You will learn the fundamental principle of some important techniques. You will able to understand the working of some important instrument which are used in forensic science investigation. Now, let us begin with introduction. Forensic science applies scientific principle, techniques and the methods to the investigation of crime. A forensic laboratory has two purposes. Firstly, it is responsible for analysis of evidence and involved in all aspects of evidence recognition, collection and preservation. Secondly, it must ensure the law enforcement officer, investigators, attorneys, judges and juries are educated in understanding the parameters which surround collection and testing of evidence. The emphasis is on research and development for refining new techniques and instrumentation, assessing the operating needs of laboratory and examining the operations of forensic laboratories and their interface with criminal justice agencies. Although the exact manner in which the physical and chemical properties are analyzed for each substance differs. The analyses are all based on principles of scientific method. Traditionally, forensic expert would analyze all types of physical evidences. Their familiarity with numerous forensic investigation methods was tremendously varied, and their capability to bring out any specified investigation was inadequate due to the knowledge and resources. Now, let us begin with the biology division, where we have polymerase chain reaction, which is also called as PCR. PCR is a technical method in a molecular biology to intensify a solitary or some copies of a part of DNA through several order of extended, producing millions of reproduction of a definite DNA sequence. PCR is applied in a forensic research laboratory and is particularly beneficial since only a small quantity of original DNA is mandatory. For example, sufficient DNA can be obtained from droplet of a blood or a single hair. Polymerase chain reaction is an important process of several genetic investigation research laboratory, sideways with the application in DNA fingerprinting for forensic and other anthropological genetic cases. The polymerase chain reaction is an influential technique that has swiftly become one of the best extensively used method in a molecular biology, as it is rapid, inexpensive and simple. The technique intensifies particular DNA fragment from small amount of a source of DNA material, even when that cause DNA is of comparatively poor quality. PCR is a quick and easy method for generating unlimited copies of any fragment of DNA. Then we have restriction fragment length polymorphism, that is RFLP method. RFLP is a technique where entities may be distinguished by analysis of patterns derived from cleavage of their DNA. If two entities diverge in the space between position of cleavage of a specific restriction endonuclease, the dimension of the fragments formed will differ, where the DNA is processed with the restriction enzyme. The resemblance of the arrangement produced can be applied to distinguish species from one another. Restriction fragment length polymorphism, that is RFLP, analyzes measures remains of DNA comprising short sequences that differ from person to person, so-called VNTRs. After taking out DNA from a sample and intensifying it with the method known as polymerase chain reaction, restriction enzymes are added to cut the DNA at a specific point. The resulted fragmentation can be arranged by length with the gel electrophoresis technology to regulate how several times a given VNTR is frequent. Then we have short tandem repeat. Short tandem repeat is also known to be STR. The human genome is filled with recurring DNA sequencing. These recurring sequences originate in several dimensions and are categorized conferring to their dimension of a fundamental repeat unit, the number of contiguous repeat units and other general length of the recurrence region. DNA areas with the short repeat units are known as short tandem repeat, as I told you, STR. Those remains selected for a forensic use usually have a tandem repeat unit of only 3 to 4 base pairs that may repetitive in the DNA molecule from a few to dozen times. Evidently, units of only 3 to 4 base pairs are extremely small. This presents both an advantage and a problem in a forensic DNA work. The advantage is that only minor quantities of even severely degraded DNA may be sufficient for forensic use. The difficulty is that a very minor tester of a very small DNA segment, the short tandem repeat required to be amplified in size for investigative accessibility and efficiency. This is completed over the application of a comparatively new technology called as polymerase chain reaction. Next we have chemistry and toxicology method, where we have spectroscopy. Spectroscopy is the study of interaction of electromagnetic radiation with the matter. It is most influential tool accessible for the study of atomic and molecular structure and is applied in the examination of an extensive variety of testers. Spectroscopy methods are much more rapid and much less time consuming. They give information which is recorded in the form of permanent chart, generally in an automatic or semi-automatic manner. The structural information gained by spectroscopic method is much more precise and reliable. They are highly reliable in establishing the identity of two different compounds. They are much more selective and sensitive and extremely valuable in the analysis of highly complex mixture and in the detection of even traces amount of impurities. With this method, continuous operation is often possible and this facilitates automatic control of process variables in industry. Spectroscopy is a common procedure that can be changed in several conducts to extract the information about the energies of electronic, vibrational, rotational states, structure and symmetry of molecule and dynamic information. We have following type of spectroscopic, one of which is atomic spectroscopic. This spectroscopic is a concern with interaction of electromagnetic radiation with the atom, which is commonly in the lowest energy state called ground state. Then we have molecular spectroscopy. It is interaction of electromagnetic radiation with the molecules. This results in the transaction between rotational and vibrational energy level in addition to electronic transactions. Then we have next chromatography, which is most commonly technique used in the field of chemistry. Certain resources seem similar but are really a mixture of material. The ingredients in a combination are not chemically combined, so consequently they can be detached over some physical procedure. Chromatography is grounded on a different passage. Chromatography technique for sorting out the constituents or solutes of a combination on the basis of comparative quantities of each solute dispersed between a moving fluid stream known as mobile phase and a contiguous stationary phase. The mobile phase may be either liquid or gas, while the stationary phase is either solid or liquid. Chromatography is the ability to separate molecule using partitioning characteristics of the molecule to remain in a stationary phase versus a mobile phase. Once a molecule is separated from the mixture, it can be isolated and quantified. The basic principle is that components in a mixture have different tendencies to absorb onto the surface or dissolve in a solvent. It is a powerful method in industry where it is used on large scale to separate and purify the intermediates and products in various syntheses. This is basically the technique of separation on the basis of relative affinity for the mobile phase. Now let us move on. Narcotics Identification Kit. The global drug problem continues to increase with the production of and trafficking of drugs and narcotics and impacting communities worldwide. The Narcotics Identification Kit field ostensible test kits are a part of tranquilizer drug identification system that is considered to speedily classify materials of being prohibited or controlled materials. The NIK system of narcotic identification is the only global customary or field testing apprehensive substances. Using the advanced chemistry, the system presumably identifies the most commonly encountered narcotic and street drugs. The NIK polytesting system is the most widely used and successful presumptive drug testing system available in the market today. Law enforcement officers are expected to combine their investigative skills and experience, knowledge of the characteristics of suspected narcotics and the expected color reactions from the field test to appropriately classify assumed narcotics. The NIK system is designed to presumably identify compounds based on probabilities and the use of multiple tests in succession. The more tests used in identification of a substance, the greater the probability of positive indication. The other divisions includes polygraph. The polygraph is an ancient and paramount recognized lie indicator technology. The polygraph is a set of equipment that accurately measures and registers numerous functional directioners like blood pressure, pulse, respiration and skin conductivity while the subject is asked and answered a series of questions. The belief is that deceptive answers will produce physiological response that can be distinguished from those related with the non-deceptive responses. In current year, brain movement has also instigated to be measured in this site. The polygraph is widely used by analytical staff in a variety of medical and scientific settings for purposes other than lie detection. In lie detection circumstances, use is grounded on the evidence that dishonesty is escorted by variation in the action dignified by the polygraph. The polygraph is occasionally known as lie detector, which is misnomer. A polygraph does not sense lies, but only stimulation which is expected to accompany telling a lie. Polygraph inspectors have no other choice than to measure dishonesty in such a secondary way, as a configuration of a physiological movement state associated to deceitful does not exist. 3 of the 4 most widespread of recognition processes by the polygraph are built upon the premise that while answering relevant questions, liars will be further stimulated than during responding control queries due to fear of discovery. Since the development of polygraph, it has been applied in criminal investigation. Now, let us move to brain mapping. The brain mapping test is done to interrupt the behavior of the suspect and corroborate the investigating officer's observation and suspect statement. It is the test that maps the brain to reveal guilty knowledge. It measures the changes in the electrical field potentials produced by some of the neuronal activity in the brain by means of electrodes placed on the surface of the skin covering the head and face. The changes directly related to the specific preconceptual or cognitive events are called event-related potentials. In simple words, it is based on the finding that the brain generates a unique brain wave pattern when a person encounters a familiar stimulus. Commonly used method in India is called Brain Electric Activation Profile Test, also known as P300 wave test. During this test, forensic experts apply unique technology to find out if suspect's brain recognizes things from crime scene that an innocent suspect would have no knowledge of. Consciously, experts say to the brain fingerprinting test as the brain mapping test is also called matches the information stored in the brain with information from the crime scene. Studies have shown that an innocent suspect's brain would not have stored or recorded certain information which an actual perpetrator's brain would have stored. Now let us move to narcoanalysis. Narcoanalysis test is also known as truth serum test. They are drugs sometimes used clinically. Some of them seconal, hyoscine, which is also called scopolamine, sodium panthenol, sodium amytal and phenobarbital. These drugs produce a state of semi-consciousness in the subject and reasoning faculty of the individual become ineffective. These drugs works on the principle of inhibiting the thought filtration procedure of the brain. Nowadays, the importance of narcoanalysis test is increasing with the time. It has great importance at the time of crime investigation. In fact, it has become an integral part of crime investigation. There are so many cases in which narcoanalysis test was conducted by investigating agencies. It is used as a preventive forensic tool to apart the planned crime, busting the conspiracies. It can prove to be valuable technique to prevent the organized crime in the hand of a competent team of experts. Now, let us summarize what we have learned in this module. A forensic laboratory has two purposes. Firstly, it is responsible for analysis of evidence and involved in all aspects of evidence recognition, collection and preservation. And secondly, it must ensure the law enforcement officers, investigators, attorneys, judges and juries are educated in understanding the parameters which surround collection and testing of evidence. The polymerase chain reaction, that is PCR, is a technical method in a molecular biology to intensify a solitary or some copies of part of a DNA through several orders of extent, producing millions of reproduction of a definite DNA sequence. Restriction fragment length polymorphism, that is RFLP, is a technique where entities may be distinguished by analysis of pattern derived from cleavage of their DNA. Spectroscopy is a study of interaction of electromagnetic radiation with the matter. It is most influential tool accessible for the study of atomic and molecular structure and is applied in the examination of extensive varieties of testers. The NIK system is designed to presumptively identify compound based on probabilities and the use multiple tests in succession. The more tests used in the identification of a substance, the greater the probability of positive identification. Thank you.